Very good afternoon, everyone, again. This is our third event of our webinar series, and today's webinar is about the transition to computational biology. We have two very special speakers with us here today, and we also have an RSG-India alumnus who will be chairing the event. And the speakers will be talking about their valuable insights and their experiences while they transition into the field of computational biology. So before handing it over to the chair of the event, I'll just be introducing RSG India a little bit, who we are and what it is exactly that we do. Are you able to share that? Okay, great. Okay, cool. So RSG India is the Indian Regional Student Group of ISCB-SC. That is the International Society of Computational Biology Student Council. There are a lot of abbreviations, but that's just how the names are of the student bodies. So the IICB-SC is the International Society of Computational Biology Student Council, as I previously mentioned. It is an international student body which has regional student groups spreading over 26 countries around the world. It aims to promote interaction among students pursuing research in the field of bioinformatics or computational biology and build a community of computational biologists so that they can collaborate and guide each other. There is more networking this way. You can call it an initiative for the students, by the students. So now RSG India aims to create a space that unites students interested in this interdisciplinary field of computational biology in India. We aim to create a platform for networking, ideation, and spreading awareness about the scope of this field. RSG India has members from all over the country. Could you go to the nodal slide, please? Okay, never mind. So I'm so extremely sorry. Do you want me to share my screen?
Please, you do.
Okay, cool. I'll do that. I'm so extremely sorry for this inconvenience. Great. So, oh no, I'm so sorry. Okay, so can you see my screen?
Yeah.
Okay, so recently we started an initiative of nodal representations similar to other RSGs around the world. So we've started setting up new nodes in various universities across India. The most recent addition is VIT Vellore. So what this does is this helps us connect with a larger number of people and consequently learn more, grow more, and as a result, perform better. So if you wish to open a node at your university, do mention it in the feedback form that we'll be circulating at the end of this event, and we will get back to you as soon as possible. So these are a few things that we do as RSG India. Now, instead of, we used to have CBDGs, which are CompBio discussion groups, but now we have CompBio cells, which is CBCs. We hold workshops, we have student symposiums, we have various scientific and academic competitions that we hold as well. Recent activities. Recently, we had a webinar of IPR and bioinformatics on December 13th, 2020. It was about intellectual property rights in bioinformatics. The speaker spoke about patenting laws in India and outside of India and how the laws make it easier or difficult for the patenting process. Next, before that, we had the webinar titled Mental Health Issues in Young Researchers on October 10th, 2020. Before that, we had a session on the topic bioinformatics and big data.
Hello. Good afternoon, ma'am. Ma'am, I am not audible.
Sumitra, you're mute.
I think Sumitra is now presenting. So let's let her finish. And she's audible, very much audible, I guess. Okay. Sumitra, please continue.
Okay. Yeah. So, So we had a session on bioinformatics and big data, the career, the scope, and the future in this field on 7th March in the IIT Madras Tokyo Tech Joint Symposium at IIT Madras in Chennai. The first student symposium that we held was titled Computational Biology, Connecting Domains of Biology, and it was held on 22nd November 2019 at JNU Delhi. We also had a panel discussion on career opportunities in computational biology and bioinformatics in September 2018 at the 17th International Conference of Bioinformatics at JNU Delhi. And then we also had a scientific writing competition, slightly different from all the events that we've had previously, in August, September of 2018. Now, these are just a few images of the events that we've held previously. As you can see, it's a scientific writing competition and the symposium. This is the student symposium that we had at JNU. This is the panel discussion at IIT Madras. And this is the webinar that we had on mental health issues. And this was the webinar that we had on intellectual property rights in bioinformatics. This is our core team. We have students working from JNU, IIT Madras, IIT BHU, Shastra, VIT Vellore, IIT Bangalore, and yeah, I think I've covered all the universities. This is Dr. Rita Sharma. She's a faculty advisor and our mentor, and she's an associate professor at BITS Pilani. If you wish to join us, you can email us at rsgindia@iscb-sc.org, or you can go to our website. You can connect with us on Twitter, LinkedIn, Facebook, and yeah, I've covered all the social media sites that you can connect with us on. Now I will hand it over to Shankar to explain the survey results. Hello, Shankar, can you hear us?
Sorry. Yeah. So in the registration form, we had asked the attendees a few questions. So regarding these subject majorly, all the attendees were from biotechnology and bioinformatics domain. And interestingly, we had a fair share of attendees from the computer science background as well. Next slide. And the second question was about their experience in this field in particular. And it looks like more than 60% of the total were upcoming computational biologists with around a year of experience. And we hope this webinar would help you make informed decisions. Next slide. And regarding the choice of programming, Python and R was their major choices because of their different libraries available, I guess. And finally, regarding the future plans, most attendees were open to opportunities in both the industry and academia. Over to Sumitra.
Okay, so our two guest speakers for today are Dr. Leelavati Narlikar from NCL Pune and Dr. Shani Das from PrecisionLife Oxford. Now I'll be handing it over to Dr. Arjun Ray, who's an assistant professor at IIIT Delhi, and we're so absolutely delighted to have him. He's also been previously a president of RSG-India. Thank you so much for chairing this event, Dr. Ray, and now I will hand it over to you.
Thank you so much. And first of all, yeah, if you could just, I'll share my screen then.
Okay. All right.
So first of all, it's such a proud moment for me to be back to my fraternity. And it was quite nostalgic to see some of the terms that earlier used to haunt us for 2 years, CBDG and all those things, and gathering students. So I think you guys are doing an amazing job. And I understand fairly well how it is challenging to keep the excitement up, especially in these times. And I think you guys are doing a fabulous job. And the topic today, I thought, was extremely interesting, since I haven't heard generally people talking about this, mostly in platforms. So for this, I congratulate you guys. And we have lined up two amazing speakers. I think, especially since I was more familiar with Dr. Leelavati's academic career, but then I started reading up on Dr. Sayoni Das, and I think my last 4 days has been gone in reading up on their papers, which was really exciting for me. I was told to say a few words, and instead of that, I thought of just making a presentation. How do I present here? I don't think I have the rights to present. Do I? Yeah, I have. Okay. So I think it was quite a. Is my screen visible to everyone?
Yes, sir.
Yes, sir.
Okay. Okay. So
Hello.
this was a.
Hello, ma'am. Sound नहीं आ रही है.
I think whosoever is this, we have to check on your end, because for others, perfectly audible. Can others, anyone else?
Yes,
Yeah, it's audible, ma'am.
Okay.
Yeah,
it's all to all of us.
there's
Yeah.
no issue.
Perfect. Or else it would have been a shame. This is a topic which is actually extremely close to my heart because I'm actually a BSc and MSc in chemistry and organic chemistry, and my starting of research career, or let's say playing with science, was to begin with was carbohydrate chemistry and natural product synthesis. And I maybe, if the experimental people allow me to say it, I started with a fair bit of experimental techniques and awareness on those things. But what got me into experimental research was first of all shiny beakers and fancy apparatus, because that was the kids thing that always was in front of me from my childhood days. Then secondly was mixing things to create something. And that was especially fascinating for me, that you could, in chemistry especially, that you mix two compounds and suddenly something else comes out of thin air, or you do experimental techniques and you're suddenly seeing DNA on your gel band. So those were exceptionally exciting for me. Then there was the challenge. And the challenge was obviously, as the Star Trek, anyone who's a fan would know, that wandering where no man has wandered before. So the challenge was for me was to find out things which perhaps was not seen ever in the human race ever before. And the multidisciplinary skill, that you had to have a little bit of background of many things rather than being completely dependent on one. Now, this was what I got into experimental research with, beginning with it. Then I got a chance in my master's to work in Cambridge University and in Tom Blundell's lab, and who's a exceptionally good lab to work with, and it changed my life towards computational field. So then I thought of how it revised my goals, and this is exactly how I think my goals got revised. That instead of, now I had fancy computers and supercomputers to play with, instead of creating things, now I was making new algorithms, designing new softwares, and analyzing new findings. The challenge obviously remains, and I think it became even more faster to look into these things. And in terms of multidisciplinary skills, I don't think I could have been in a better field, because this is a field where I, in my research, deal with physics, chemistry, bio, maths, statistics, computer science. And I've written a scope for artistic expression, because anyone who publishes regularly knows that making figures beautiful is also an artistic expression. So that also keeps me excited. And to end with it, this is where I always tell my students that I don't know where I'm heading, but at least I'm on my way. So with that, I'll stop talking about myself, because today we have, first of all, I would like to introduce Dr. Leelavati Narlikar. Let me just stop my screen share. So while she sets it up, let me just give you a brief introduction about her. Dr. Leelavati, she obtained her bachelor's in computer engineering and then did a PhD in computer science from Duke University. She did her postdoc in NCBI in Bethesda and then joined Pune University, NCL Pune as a Ramanujan Fellow. Then she was a Wellcome Trust Fellow, and now she's a scientist at NCL Pune. Our research area is relating towards machine learning and computational biology, though I think a better way to describe it is that she works exceptionally well on various cellular biomolecules, gene expressions, developing new statistical algorithms. She regularly publishes in extremely reputed journals like Nucleic Acid Research, Genome Research, and all these. And what was fascinating also was that her lecture videos on YouTube, and those were some of the things that I even went back in these couple of days to learn some of the new stuff. So with that, I will hand you over this platform, Dr. Leelavati, and thank you for agreeing to talk to us.
Thank you. Thank you so much for the lovely introduction. And thank you, Manisha and Shruti, for the invitation. I'm really happy to be part of this. Let me just share my screen. So I have myself benefited from ISCB. As a student, I was got some travel funding from them to go to conferences and things like that. So I'm very happy to be part of this. So at that time, I don't remember there being any of these regional groups, and I think it's great that you have these. And I'm embarrassed to know that I didn't know about these until until Shruti wrote to me. So while I'm thrilled that you have one, I'm going to do my bit and advertise for you, because this is great. This increases our presence in also in the all the conferences that ISCB organizes. That would be great, because I always feel that the Indian community is not represented enough, although they're doing such great work at these places. Of course, there are many reasons. These conferences are not necessarily held in India, and that brings up different problems for us. But even then, right? So it's great. It's a wonderful initiative, and thank you for having me on. So I'm supposed to talk about my journey, my hurdles, and what I've learned over my career. So I'm going to do that. But before I can put that in perspective, let me just give you a brief, sort of a few slide stuff about what I do. Okay, so hopefully that will, like Arjun said, that I've done a PhD in computer science and things like that. Maybe that'll make a little more sense when I tell you how I got there. So my lab is, you can see my slides, yes? And you can see them in the slide.
Yes.
All right. So my lab is interested in developing machine learning algorithms to solve problems in biology, specifically molecular biology, and I'll give you a couple of examples.
But still, you are in the first slide. Did you change the slide?
I did.
Oh.
So you can't see that. Okay.
No,
all right.
Let's
see what's going on.
So it's changed now.
Is it changed now?
Yes.
Do you see the arrow, the fancy animation?
No.
See the oval shape.
I think you need to share the entire screen instead of just presenting.
Oh, does that work better?
Right.
Okay. So let me just do that for a second. A minute, huh?
Does this happen to you also? Do you have to do it too, normally?
Yes.
Okay. Okay. Is it too bad? Wait, hold on.
So when you click on present now, there will be one option of your entire screen.
It didn't give me any option right now, so I'm just.
It should be on Zoom. In the Zoom options, there should be.
So this can you see now?
Yes.
Yes.
And do you see the arrow?
No, just
No.
You don't see the arrow here.
Okay, all right. Let me stop sharing again. Okay. So now you say I should go to the Zoom.
Screen share option. In the lower panel, you'll see screen share options.
I should probably
If you
show all options.
and there should be one selection which is show the complete desktop or show all or something else.
Yeah.
It is show all windows.
Now I have said that. Let me see.
Ma'am, the arrows will automatically come and we'll select the other. It's animation you have made. We can see your slides.
Yeah. Okay.
Now it's visible. Now it's
Okay.
visible. Now it's visible.
I don't know what
Perfect.
I did. Okay.
Hallelujah.
Yeah, I don't know what I did, so I don't know if I can repeat it. So anyway,
But yeah, it's working. Yeah.
it's okay. Yeah, right. And as you can see, it's not very fancy. It's just an arrow. But
No, that's okay.
I like to make it.
Yeah, ma'am. This was the animation. When
Yes.
you will click the next button, it will automatically come.
Great.
That's why.
Yeah.
Yeah,
Okay.
and arrows love arrows, by the way. Don't underestimate them.
Okay. All right. Great. So, right. So my lab and I are interested in developing new algorithms to, solve problems in biology. And that's what we began as. But over time, we've realized that some of the stuff that we do, right, some of the problems that we solve and the approaches we take are in fact fairly general because we make very few assumptions about the biological problems. So we think they also sort of can be applied to other domains as well. So it becomes a little more generic and more fun exercise. And so I'll just give you a couple of examples. The big question we are interested in solving is to be able to decipher architecture of the non-region. Okay? So I know this crowd has a good background in biology already. So I'll just very quickly go through this. So you have these regions which we call regulatory regions, which don't necessarily code for genes, but they affect the transcription of some target gene. And we are interested in being able to understand the architecture of these regions to a point where if there's a mutation, we should be able to tell what effect that mutation will have or not, whether it will have any effect or not. Right? And this is not an easy problem because unlike genes, which have a better structure and well understood structure of proteins and things like that, these regulatory regions don't. So regulatory regions come in different flavors. Okay, so these are just a few of them in this picture. The gray thing is the DNA, and they are named. The regulatory regions are named based on the function that they're perceived to carry out. Right, and these colorful blobs on top of these regions are transcription factors. Right, these are proteins that bind DNA in a sequence-specific manner. So naturally, if you're looking to see whether mutations in these regions are likely to cause misregulation, you should be looking at these regions. Yeah, these are the transcription factor binding sites where the protein is making contact with the DNA, right? And these are really tiny contacts, which is why the problem is hard. For example, this blue protein might bind to this little region. Also, the blue protein doesn't always bind to that region. It's going to allow for some variation, and so you need a model. So here's where computation comes in. It's been around for a long time. It's a logo kind of a representation, which I'm sure you've seen. It just represents the information contained in the a binding specificity of the protein, right? So it's just telling you that the CACG is very strong, and it's going to allow for a few variations in the downstream of the TG region. Yeah. So we are interested in being able to find these little pieces, under what conditions they are active and so on, so that we can then start piecing together how mutations in them could be causing misregulation. This is important because lots of diseases are, blamed on misregulation of genes rather than mutations within genes themselves. Yeah. Luckily, right, we have these really smart biotechnology people, right, who are generating lots of different data. They're doing these clever experiments that can probe on a genome-wide scale various different biochemical activities, right? So you can identify now in one shot across the genome where a specific protein might be binding, where there might be an enhancer mark. So the enhancers are regions which I showed you in the earlier picture, regions which are open, right, open for binding and things like that. So let me. So this has been around for a while, right? And typically, when you have such data, the downstream processing proceeds as follows, right? So this is a common approach that's taken, which is to do some sort of an average analysis, right? So so you come up with things like, I suspect that my protein is organizing chromatin in a certain manner. So let me just align all my protein. This is a CTCF protein binding sites. Let me align them. Okay, so that red curve that you see is telling you the signal across maybe thousands, tens of thousands of CTCF binding sites. And then let me just, find out how the nucleosomes are arranged or the histone modifications are arranged, right? Or how their signal looks. And you get a plot like this, right? Or you might say things like, here's where where I see these are all the regions where my protein seems to be binding through an experiment. Let me do a motif discovery thing, and this motif is enriched and so on. So our our rationale is that, is this the best you can do? So imagine in the CTCF case, right, in the left side, maybe you have these nucleosomes arranging themselves in only one direction, and the other direction is like noise. But if you have lots of these CTCF sites, some of them are flipped around, half of them are not on the other side, so you will still get this picture, right? Or maybe just 80% of the CTCF sites have this a cool arrangement. What's going on in the other 20%? So clearly, we don't really expect. We know that there's not a single sort of signal in this data, and what we are trying to do is to sort of go around saying that we don't know what to expect, but we do assume that a single room doesn't apply to everything. And now that we have all this data, can we learn different rules maybe from the data itself? Okay, so and our goal is to answer this, is to develop new techniques which will hopefully give new insights in biology. Yeah? So let me just give you a quick example of a map where you have transcription start sites mapped all over it. There's lots of experiments that do this for you. So we are basically targeting the 5' region of the RNA molecule and sort of amplifying that, and then you go back and look around the genome, right? So this is to look at the core promoter regions to see where your what signals are present over there. And the data looks like this, right? So you're going to have these peaks all along the genome. If you want to look at the core promoters, then you need to look at the regions around it. And the typical, approach is to just align these regions like this. Yeah. So here, what I've shown you is an image plot of all the promoter regions aligned based on the transcription start site. So the middle is the transcription start site. I've gone up and down 50 base pairs, I think. And each row that you see in color, 4 colors, are the ACGT signal, right, at those regions. So it's the DNA signal, DNA at those regions. Right? And typically when you get such data, you also see the data box out there, don't you? So you take the data box and you scan it, and then you come up with these plots that say that, Tata is sort of enriched here, and then you have all these other motifs which we know already exist, and you see whether they are there, and then you sort of come up with a average plot saying that, yeah, 30% of the regions have Tata and so on. So what we did was said that we don't know. Let's assume we don't know nothing. Let's say we know nothing about which elements are present. And so we developed a machine learning algorithm, like unsupervised learning, really clustering, except that we also do feature selection. And you can ask me these questions later on, the technical details. But you see that there's a lot more diversity in the data. And there are many regions that don't conform to Tata box and all this, and you can explain them with other signals that you see here. Yeah. So, so there's another quick example where we're looking at open regions, and so open regions are open because of many different reasons, right? They could be enhancer regions, they could be active promoters, or some other reason completely. And the hope is that if you identify these open regions using DNA's hypersensitivity assays or ATAC-seq, if you do this, then you're going to get in one shot all regions that are presumably doing something, right, which are doing some regulatory activity. At the same time, we do know that it's going to be a diverse set, right? So normally people will do a motif discovery method. You apply meme or Homer or something where you will look for enriched motifs, right? But when you apply these methods, you're again saying that all the sequences are going to be similar, right? And then trying to get a signal from these. So what we said was, let's assume that we know nothing again, but we do know that you have only a set of transcription factors that are active. You also know that the sequence activity is driven by a combination of motifs. And you also know that the experiment is reporting a mixture, right? You've seen that it's reporting diverse regions. And so we developed we hypothesized a model that says that let's start off with our sequences, right? The regions that are reported by the high throughput experiment. And what we want to do is to sort of reorder them, split them into different subtypes, where every subtype is going to have an enrichment of a set of transcription factors, right? So those colorful things are the transcription factors, and those black blocks just say, the black dots say that that motif or the binding site for that protein, which looks something like this, right, is going to be present only in that set or enriched in that set with some probabilities, right? So we say that you have all these set of motifs that are active. You have modules because you have promoters acting differently, enhancers of one type acting differently, silencers behaving differently, and so on. But we have a lot of data. So we're going to say that although you have modules, we expect enough representation of such types. So you have enough promoters around, you have enough enhancers of a certain type, and so on. But they're going to have these combinations of protein binding sites. But what you're getting for the experiment is this complete mixture, right? You're getting only the green regions. So we developed a method that tries to get back everything. And this is inspired from a technique in computer science, which is called topic modeling. So the idea is that you have lots of documents which you don't have time to read, but you automatically split them into topics. In this case, it's modules, okay, things that are behaving differently, regions that are behaving differently based on words. Now, in topic modeling, you already know the words because you know the language, you know English or whichever language you're doing the clustering into topics in. But here you don't. So you're also learning the motifs. Okay, so it's a mega learning process. But it's sort of something that kind of works. It's not yet published, but it's available if you're interested. So overall, right, if I had to just say what we do, we are interested in developing new methods for new technologies. So these are all these technologies that keep coming. Every year you have some cool method that identifies in one shot, one type of regulatory region, set of things. So that's essentially what we are interested in. And some of the work which I have not spoken about is shows, we've looked at, so for example, the promoter stuff, right? That data was DNA, but that method can be applied pretty much to any kind of data. It doesn't have to be a DNA sequence data. So how did I arrive here, right? So this is all the science that I do. And so when Shruti wrote to me, it made me think as to why am I doing what I'm doing, other than the fact that it's a lot of fun. So this is my super narcissistic slide, right? So it's all about me and my journey. So I have, like I'm sure all of you have been, influenced by several people, right from when I was a young child. So let me, so this is what I did. I went and wrote down what, this is really after she made me think about what is it, what gyan can I really give? Not much gyan here, but it made me think. So I just sort of split my life according to different time points as to what I might have learned at that time. So very early on, right? This is, I think, in middle school. That time, my influencers, influencer now means something very different. And these are not those kinds. These are real physical people who I have interacted with. So were mostly parents and teachers. Okay, and I realized, so I had really good teachers to teach me maths and science, so that I enjoyed doing. But I should say here that I come from privilege. Okay, so I have been lucky enough to have good teachers and good schooling, but I also come from this academic family. So my family is filled with academicians, and my parents, my sisters, they all, my sisters at that time when I was in middle school were not doing science, but they are now. But my parents definitely encouraged us to solve puzzles. Okay? And these were just regular puzzles which would need you to think a little bit, some logic, but some maths that a middle school or high schooler could do. And this was a lot of fun. Okay, and this is also fun because we would do this as an activity, right? And we would be competing against lots of people in the family, so parents and sisters essentially, right? And I'll come back to this point. But that's essentially what I learned in my middle school, that I like maths and I like to solve puzzles. Okay, then things changed in high school because parents had very little influence on me and was mainly peer, right? And I hung out with people who said that doing maths was good, doing engineering was what one must do. Medicine is not good. And if you do biology, it's just mugging up stuff. So let's not do biology. And I said, yeah, you're right. I don't like biology. And that was my conclusion. I think after 10th standard, the first opportunity I got to completely remove biology from my curriculum was after 10th, and I took that opportunity. And then naturally, because I had already decided that what I wanted to do was engineering, I got into an engineering program. Again, my peers were a big influence, but this time my sisters were as well, because they're much older, and so they knew kind of what research or what science was. And they encouraged me to, pursue, since I was already in a computer science program, to go, and do problems and algorithms. And I really started enjoying at that time, because I think that it still connected me to puzzles. So writing an algorithm is still like solving puzzles for something. At this point, after doing my engineering, I didn't really want to take up a job. I didn't know what I wanted to do, but the only option if you don't want to take a job is to do a PhD. Because my, so master's was not an option because my parents said they're not going to fund any more education. So I have to fund it myself, and master's is not, you can't, it's very hard to get funding for master's. So PhD seemed to be the only option for me. Again, I would like to stress, this is because I was I had the freedom to make that decision. I know many people cannot. Many people sort of have to take up jobs or are under other constraints. But this is about me, right? So this is what I did. I went to do a PhD, and I still had that mindset that biology is not something for me. But in the very first semester after I joined Duke University, we had to take, I think, 3 courses in the first semester, and the first 2 were core CS courses, which I enrolled for, and then I was trying to see which one to take. And I saw this algorithms in computational biology, and the seniors said it's a good course to do, so I took it. And I was taught by this person called Alex Harteming, and he was he still is an exceptional teacher. He encouraged me to read up on whatever biology I'd missed on for so many years. I also took one undergraduate course in biology in the university because I had the option of taking those courses there. Eventually he also became my advisor because he just convinced me that this is an area that has so many opportunities and a lot of fun, because it was still like solving puzzles. So what I really learned, right, was that the reason I think that liking this part about solving puzzles got me to pick this career choice is because I realized at that time that it is highly non-hierarchical in nature, right? So we were competing with parents, the 3 sisters, and anybody could come up with an answer. Anybody could come up with a more elegant solution than the others to these were like trivial puzzles. Yeah, I mean, it's not like super fancy or hard puzzles, but these are things that just made you think and apply logic in a different way, maybe. And that's when I realized that people who often come from different backgrounds think differently, and they might be able to solve the problem in a better or a different way. And so I'd like to because I know there are lots of first-year students and second-year students here, students who have not decided what to do. It doesn't matter if you don't take computational biology. Whatever you decide to do, if you're doing science, please do remember that it is highly non-hierarchical. You can come up with a solution, but a much better solution than your advisor. And chances are you will. But unless you speak up, you're not going to be heard. So make sure you speak up. You make sure you're asking enough questions to come to a point where you can come up with a solution. So also, right, this is a little reassuring that whatever decisions you take in life, none of them are irreversible, or many of them are not. Okay, so when you are at a crossroad thinking, should I take physics or not, just go with your gut. So maybe later on you realize that doesn't work out. You can come back and revisit it. So it's not that. Don't take too much stress about these things. You just go and you can always come back. I also realized that I got a taste of research. So those puzzles were where you already knew they could be solved. Right? So my final year project in my engineering was something that introduced me to data structures, and they gave me a taste of research. And I think that is really why I didn't want to do a job, because I thought that a job which is not probably true, but that's just what I thought at that time. And then what Alex taught me was the value of being, of having a fantastic teacher. Can completely change your outlook, can introduce you to totally different things that you didn't think were worth looking at, doing, and things like that. Right. So this is my sort of analysis about what I learned over these few years and why maybe I'm doing computational biology. It's just because I've had a great teacher, who convinced me to do this. If this teacher had convinced me to do input-output algorithms, which is what I had decided when I joined PhD to do. The good thing at the university was that you didn't have to choose your area of research or your advisor until at least one semester, usually 2 semesters into the program. So that was actually useful because it let me explore what I wanted. If there are questions, you should just stop and ask me, but I don't think there's anything worth questioning at this point. But regardless, right, if you have something to say, feel free to interrupt.
I think one question just came up is Bodhayan asking how to find fantastic teacher.
I know. I wish I knew. So I think one thing is that if a teacher is good, you will hear that the person is good, right? So you will get feedback from people, and I would say take their courses, even if the course is something I don't know. I mean, for me, if it is quantum chemistry, I'll do it. If it is something, if it's a teacher who really teaches well and has good ratings, so to speak, right? So do it. I mean, especially when you're in the university, if you do have the option of taking courses, multiple courses, different, so do it. Unfortunately, our system doesn't always encourage that. Because we don't have those many universities. You have these institutes, you have engineering institutes and things like that. So it may not always let you explore, but now you have lots of things online, so you can do that, right? So there's some snags, right? Some hurdles which which also I took some time to jot down. Is that you need to, it looks simple that I wanted to do puzzles, and then I decided to do computational biology. I had a good teacher. Well, that teacher was there for that semester to teach that course, and of course afterwards he was my PhD supervisor. But you still have to learn the language of these other areas, which you may not be well versed with, right? So so for me, because I come from the CS background, is how many data points do you need? How many experiments do you need to conduct? What were the assumptions made in that experiment, right? So all these are very important things that you really need. This is where the hard work comes in to get there. So how do you do this, right? You have to read a lot. There's no easy way out. You have to read research papers. You have to read textbooks. And many of the textbooks, by the way, especially the biology ones, are being rewritten. But it's a great idea to read whatever is current right now and then read the papers that are associated. You have to interact with other folk from other areas. Sometimes, like I had done, I had a preconceived notion about certain areas. And the sooner you get rid of those and start respecting all areas of science, the easier it will be to get to a level where you can make a meaningful contribution, right? So, if I've heard, from both sides, if you're a computational person, there's these things that, they're just working in the lab, mixing things or whatever. So that's kind of a dismissive way of saying that it's not as important. And similarly, I'm sure experimental people people who do experiments have similar things to talk about. So by the way, I got an opportunity to work in a lab. This was like way when I was really old, so during much after my PhD, because I had written a proposal which said that I would do this, so I decided to do it. I went and spent 2 months in an experimental lab, and the postdoc there was mentoring me, and we were trying to clone something. And that was a real eye-opener for me, because I realized how much work really goes into the whole thing. It's not just mixing things, right? You really have to put in a lot of your brain cells and a lot of course, hard work. And that's also when I decided that it's good to know what experiments are done and what the limitations of experiments are, of specific experiments. So it's good that I did that 2-month thing, because I know what goes into it. I also know the noise things, sources of noise that get into it, but I also realized that is not something I want to do. Okay, but it was an informed decision. So do that. So if there's an opportunity to do it, make informed decisions along the way, instead of just hearing people and basing your judgment on that. And then when you now it's easier to listen to talks. I've also realized that we as a community, student community, doesn't always ask questions. I think having an online forum like this one, or now there's so many of them, just register and listen to these talks and ask questions. There's this kind of anonymity there, right? So you can ask questions, and you don't have to feel shy to ask questions. So you should do that. And you'll realize that many people might also have that question. So don't worry about it. And of course, over time, you should also give talks to people of other, other domains, coming from the other domains. So if you're a computational person, give talks to where you have more experimental people, because the questions they ask are going to open your eyes, right? Because they're going to have different kinds of questions. So talk to many different diverse groups in the process. And this is something that I wish I had been told and gotten me very better prepared. You're going to get lots of rejections, okay? And for a long time, not for a long time, for the first 2 papers that I wrote, I didn't get rejected, right? It went through maybe through one revision. And I was boasting about this to my to a brother, my brother-in-law, and he said that I expected him, to say, "Oh, that's great," and he said that just means you're not trying at the right places to submit your. This is for paper thing, and that that was really good to hear from somebody, right? Because the person is saying that you have to get rejections. If you don't get rejections, then you're not trying high enough. So that I think is good advice. And after that, of course, I got so many rejections in papers and grant proposals, and you are going to get them. Okay, but it's a good idea to know right now how to react to these rejections, is what I think. Because especially in our area, when we are working in an interdisciplinary thing, you need to understand who the audience is going to be. So it comes back to learning the language of the other area. But if you want the other person to praise your work, which is what you want to do, right? When you're writing a paper or a grant proposal, you have to write in their language. So that is something that you have to learn. So you need to know who your audience is and figure out how to write for them. You have to write for the reader, not for yourself. At the same time, you should not get bitter. I mean, you are going to get bitter, but not you need to get out of it. I have been through stages where I think that this committee was bad. So maybe yes. So maybe the committee was bad. But why was it bad? Was there something about what I presented, or what I wrote or something? And try to get thinking. Don't think too hard about it, because that will make you more, get you more depressed. But just try to take it as a positive. And it's okay to get rejections. Okay, you are going to learn from them. You'll get better. In fact, when you're writing and your advisor is the last author, you won't feel it as bad as you will feel when you are the senior author, and which will happen at some point. At some point, you'll also realize that the system is bad. You want to change it in some way. And you will think that way, because the system is kind of old, right, all through. And our field is very new. Yeah, and it keeps changing, right? You have new methods, you need to have new techniques, and you need other new things. If you're writing a grant, you want to have some flexibility, because you don't know how your project is going to go. And you might, at this point, so I have a research group in India, although I didn't do my PhD here. But I've realized that students don't get a chance to go and present their work in international conferences as much as they should. So I think this is a system that needs to be changed, right? And you need to sort of get there and do it. And sometimes you'll realize that whatever you're thinking of doing will not benefit you, but it will benefit others. And I'll tell you an example of completely unrelated to computational biology, but where I benefited from somebody else having changed the system just before me, which was when I joined as a Ramanujan Fellow in NCL, and I was already expecting my first child. And there was no daycare in there, in the institute. And I heard there was this committee that had been trying for a couple of years to get a daycare set up, and they were still at it. They asked me to join their 1 or 2 meetings of theirs to give my inputs. They took my inputs. Afterwards, I went on maternity leave. I came back, and the daycare was out there just in time for me to send my child. And these people who were trying to get the daycare set up, they had been trying for a while, and their kids were now in school. So they didn't really benefit in the way I benefited. But they realized that these scientists, these are faculty at NCL, and they realized that even if they didn't immediately benefit for themselves, they were, what they did was actually helping lots of graduate students and people like me who needed a daycare and could help out the institute, right, by sending our children to the daycare just on campus. And so I met one of the committee people recently. The chair of the committee at that time was a person called Venkat. And I mentioned to him that, it's all your hard work that we managed to send our kids, and you really didn't benefit from this. And he said that that's not true, So he said that for 2 reasons. One was that when they started this whole exercise, none of the institutes in the immediate neighborhood had, or at least the big institutes had a daycare on site. And what happened at the end after NCL got their daycare, these other institutes like IISER Pune, IITM, and CCS managed to, use this as a precedent and tell their administration that, this is what you do. This is what has been done, you can do it. And so they, got that activated. The other reason was that he said that in the process of setting it up, he ended up speaking with lots of daycare providers, lots of other scientists, lots of institutes. He understood many types of business models because these were daycares that were set up. And that actually seems to have helped him when he started his own startup. So now he's not at NCL, he's started his own company. And he says that he learned from that whole experience. Although his daughter, didn't really go to the daycare as much as they would have liked to early on. So that's something that, you should think about. That something that you think that should be changed will be useful for the area, the domain, and then see if you can do something about it. The other thing was that when they did this, this is important. So when you're trying to change the system and propose a new system, you can't just say, I want to change. You need to propose a new one, right? The changed one. And that was done really systematically. Just like how you would write a scientific paper, right? With points, with why do you need this? What are the, government mandate? You have so many people, scientists working, you have so many graduate students, you have so many kids in the system, right? With parents working here. And so they had this whole setup where you really could not argue, right? And that's how they got it done. Right. This is an advice that I try to take to heart. It's not hard for me because there's lots of people out there smarter than me. So hang out with people smarter than you, even if they're not in your area. Okay, because who knows, maybe right now you speak with somebody who does physics, and a few years down the line, you think that there's something from, you can apply from nuclear physics that you had heard to this problem that you're trying to solve. So just keep talking to people smarter than you, and also, with integrity, of course, but who will sort of help you give you diverse ideas and different perspectives. Especially because we are in an interdisciplinary area, and many of the big discoveries happen because somebody is thinking from a completely different point of view, right? And tackling the same problem that has been looked at for several years. Yeah, so I've come to the final slide now. I think this is a broad sort of slide where most of us are trying to use math or computation for natural sciences. or taking inspiration from natural sciences to have a new theory in maths or computation. It's if you're doing it the first way, that where you're trying to solve a problem, it's just in some sciences like biology, chemistry, whatever, and you're trying to apply some math or computation or machine learning, some tool that's already available. Here's just one last piece of advice, is to ensure that you really understand the tool that you're applying. Okay? To a point where if asked, you should be able to code it. Right? So you understand really what is going on in the tool. Even if it's a negative result that you're getting, it would be really nice for you, very satisfying for you if you figure out why you're getting a result, right? Instead of just saying the method didn't work. But it's always really extremely satisfying to know why something worked or why something didn't work, even in computation. Right? And similarly, if you're trying to, if you come from the math background and you're trying to model a system, you really need to know the assumptions made by the person or the people who were generating the data, right? So it works both ways, but you really need to know. Otherwise, you fall somewhere in between, where you're neither developing a new technique, nor are you solving a problem in a new way, right? Or getting new insights. So make sure you don't do that. And finally, I think we're all lucky that you're doing computational biology or computational chemistry, whatever you guys are doing, because given the situation right now, we should count our blessings, because we don't have to go to the lab. Many labs are closed, so in that sense, our work doesn't get hampered as much. So put that to good use. Also try not to get too, it's easy to say, but situation outside is bad. And the more news you read, you're going to feel worse. But you're in a state where you have a computer, and hopefully you can access bigger computers back in your lab, and keep working and take your mind off these unpleasantness that is happening. And I'll stop here. I think I might have already gone over time. I have. I'm sorry.
Thank you so much. And it was so exciting to hear you. Actually, there have been tons of questions coming in, and I didn't want to stop your flow. So if you could answer a few of them which have come across. So first of all, one question that I would like to ask from, I think from my side, is that how did you find communicating with experimentalists? And this I understand from both sides. Experimentalists have a different language, a different requirement, a different way that they see science. And you, from a computer science point of view, and a computational biologist point of view, have a very different way we see things and we ask questions. So how easy was it to have this bridge between understanding what they're talking about and what you're trying to ask?
That's a good point. It is hard. So for that, I would like to thank both my PhD and postdoc advisors, because they would take me to these meetings, right? And you would follow the lead to figure out how to talk with them. And even in group meetings, when we would do group meetings without the experimentalists, right? Because we were a completely CS group, computational group. There was, although there would be questions about how do these people do the experiments, or why are they not doing it this way? There was this really code of respect, that if there's something that you don't understand what an experimentalist is doing, it's better to go and ask directly instead of trying to be dismissive, although that comes more naturally. The other thing is, I think you just have to read papers that are trying to solve similar problems, don't you think? Because then that gives you a sense of how they are, how the paper is written, and you see the model that they have used and related areas, but not. That's what I think. But it's not an easy thing. It's not easy.
Right.
Yeah.
So Dr. Barkha has asked, can even after PhD, can someone switch their research into computational biology?
Yes, I'm sure, unless your PhD is in, I don't know, in history or something. I mean, what's the PhD in?
I'm guessing it must be in science.
Yeah,
One of the
yeah.
sciences,
I mean,
I guess.
history also maybe. I mean, depends on what you've been doing. But yes, I would say yes. But it needs a lot more reading and talking with people.
Right. So someone is saying that I'm a final year PhD student purely from biology background, don't know any programming languages. Now I'm very interested to work using bioinformatic tools. Is it possible now to learn programming languages in this
Very
stage
much.
where
Very much, because the tools are, in fact, if you learn a language like Python or R and you see a tool that's written in those languages, you'll see how straightforward it is to write your own tools that do the same thing. And I think that will give you more confidence, because there is nothing exceptional about the tools out there. You know, you just have to learn. It's a language, learning a different language once, because the science part of it, anybody with a little bit of logic can put it in. Yeah. In fact, if you're a biologist right now and you have time because you cannot go to the lab, I think it's a good idea to do online.
it's the best time.
Yeah.
Then somebody asked that, is there any textbook or course for understanding problems in molecular biology or genomics that can be solved with computers?
So there are books, the textbooks that are used when you're teaching computational biology courses. You can look them up. So it depends on whether you want to do sequence level stuff. There's the Eddie Durbin, very famous book that does algorithms for those. There are also theory books. Yes, I think if you search a little bit, you will get enough. I think the best way to do this is to look up courses that are taught by top universities and see what resources they use. That's very telling.
I'll paraphrase one question. The last question is, somebody asked about any books or websites that helps improve a person's problem solving skills. I would rather ask this on a different way. Do you yourself, even apart from biology, sometimes wander off to do some problem solving, these puzzles or anything? Do you have the time to do some problem solving puzzles?
Yes.
So people are asking, which what would you suggest, if there are any? Even if that's Sudoku on the newspaper, that could be one problem solving way.
Yeah, but that sort of doesn't give you the opportunity to explore different logic-based problems.
Right.
So
what problem solving?
So I think if you go to do a search for interview questions for IT jobs and things like that, maybe Sayoni can tell because they must be interviewing people.
Yeah,
and they must be asking such questions. But I think everybody really is looking for people who can solve these puzzles. So do it, and it's incredibly a lot of fun to do them. When you get the answer, it is so yes, I think, and we still do it. So I have 2 kids, and yet I'm just waiting for them to grow a little more so I can discuss with them more than what we can do now. But yes.
Great. Thank you so much. And this was fascinating to hear. And this is a good segue for me to introduce Sayoni, especially since now we heard from a computer science background turning into a computational biologist. Now let's hear from a biotechnologist turning into a computational biologist. So first of all, thank you, Dr. Leelavati, again. And it's my pleasure to introduce Dr. Sayoni. In her words, she's a computational biologist with a background in protein informatics and biotechnology, experienced in multi-omics technology and data sets, and works on potential clinical impacts. To introduce her further, she did her BTech and MTech in biotechnology, then transitioned into doing her PhD and postdoc in computational biology in UCL, where she worked in the most one of the most esteemed labs in protein structure families in Christina Orengo's lab. And then she joined as a senior bioinformatician in PrecisionLife, and within a year and a half moved the ladder to be the head of bioinformatics now of PrecisionLife. With that, we are fascinated to hear from a biologist's point of view how a transition to a computational biology field works. Over to you, Dr. Sayoni.
Thank you, Dr. Arjun. And I would like to thank RSG-India for the invitation to speak today. It's amazing to see that the RSG group in India has flourished so much. I wish I had the opportunity of joining such a group when I was trying to do my transition. But thank you so much for the opportunity, and thank you everybody for making time on a Sunday to attend the session. Also thanks to Dr. Leelavati for sharing her experience. That was really fascinating to hear, and yeah, it was lovely to hear about the genomic network prediction as well, because I'm a biologist. I do not develop all the methods, but I really end up using them. So it was really nice to hear about that. Thank you. So what I'll intend to do today is share my experiences about two transitions that I've made. One was transitioning from biotechnology. Can everybody see my slides?
Yes.
Okay.
If you could just change your slides, because that was a problem earlier. Yeah, it's okay. Perfect.
So what I was going to do today is share my experiences of the two transitions. One was from biotech engineering to computational biology, and the other one was from academic research to industry. So I like to call computational biology data-driven biology, because that's how I got motivated into joining computational biology. So I'll not go into the details of all of the research projects, but I'll try to ensure we have enough time for questions at the end. I wanted to start off by highlighting that there's never been a better time to be a computational biologist. So we are living through a revolution in biology. We have a huge amount of data that is being generated due to the advances in high throughput biology, because of which we are getting to know more and more about the complexity of biological systems. And there's a huge drive towards understanding biological systems in its entirety, and not segregated fields that I had learned during my coursework, which is genomics, transcriptomics, and proteomics. People are trying to make sense of all of them together, and which is very exciting for biological sciences. And of course, this requires interdisciplinary research, not only from biologists, chemists, physicists, but also from computer scientists, engineers, and all different kinds of fields. We have actually in our company got inputs from marketing team as well, because innovation can come from any different place. If people want to solve problems, that same problem solving skills can come from anywhere. Also, there's more communication and collaboration between different organizations. More and more consortiums are being developed, where you have active collaborations between academia, industry, and public health services, who make use of the information that is being generated and transform them into something that can have an impact in patients and people's lives. In the next slide, I wanted to take a step back and just remind myself how I got into biology and why I got interested. So my interest started when I came to first know about the Human Genome Project in high school, and when I actually visited the Wellcome Center when I started my PhD. It was amazing and inspiring to see the entire volume in hard copy. So this here you can see the entire Human Genome Project printed out in text, and the top picture basically shows the amount of information that is there in each page. So that is incredible. And to think of the amount of genomic data that we are producing every minute, we need computational approaches to try and make sense of that, so we can actually utilize that information that we are generating. To also appreciate the developments that we have made, I have also put together a picture of slide that I got to see from Professor Jeff Barton. Here it actually shows how multiple sequence alignments were done back in 1984. So what people used to do was put the letters in squared paper and slide them over to see where the good match lies, which is incredible. Like, and they also said that they use scissors to account for insertions and deletions. I'm immensely grateful for all of the people who have done research in that area so that we don't have to do anything like that to try and analyze the huge amount of sequence data, both genomic and protein data today. But that is what basically computational biology to me is, trying to make sense of all of the biological data which is out there. And the biological data can be in different forms and it can be utilized in different ways. What I focus on is trying to solve a problem that I have at hand. And I started using computational tools to do that. So that's where my transition lies. I'll take you through my academic journey, how that happened. So my interest in biology led me to do degrees in biotech engineering in Kolkata. Soon after that, I realized I started questioning every experiment that I'm doing because of obviously the amount of time it takes and the amount of failures that you get when you're doing the experiments. I started asking questions. Why am I purifying this protein? Why am I trying to crystallize this thing which would take 6 months? Is it the right protein? And why am I testing the function of this protein by doing only one test when I know proteins can be multifunctional? So I started thinking about things how I can improve my experimental designs by making use of the data which is out there. So that's basically where my transition happened, that I realized that I wanted to make good decisions. And in order to do that, I needed to analyze the data that is available to me. I actually applied to my MS in research in IIT Delhi because I had heard about the supercomputing facility there. But sadly, after joining, I realized that you don't have a choice of too much choice about which project you can get. So at that year that I joined, I realized there were no computational projects. So that was a sad thing, but never mind. I had already joined and I decided I should continue and finish the degree because I had another very interesting problem to solve. I worked on magnetotactic bacteria. In case you've never heard of them, these are really cool bacteria that if you grow in a test tube and put a magnet in front of them, they are attracted by it. So that was a really interesting thing, but I knew that my interest lied in computational biology. So I'll just lay out what kind of experience I had. In my bachelor's in biotech engineering, there was a 6-month course where you do basic coding in C and Perl. I had done some coding in my class 9 and 10, where you do take up computer science and did C++. That was a very old version of C++, which is changed now. But that was my coding experience, very, very basic. But what I tried to do during my master's was try to become aware of what biological data is out there, just based for my research, just as a background thing. When I'm doing literature review, I'm also looking at, are other people looking at something else? And just looking at the papers and doing my bit of research. When I started applying for my, that was the time when I knew I definitely wanted to be a scientist and do research, but probably in data-driven biology. So what I tried to do was apply for computational biology positions during my PhD. And it was challenging for me to try and show competence on paper, and I can understand some people might have that hesitancy to try and do the transition, but I would encourage them to highlight your interests and efforts. Just start looking at courses, just start doing attending webinars like these. These are very informative. And if you can show your interest to a prospective supervisor, they'll be impressed by that itself. You'll frequently get these questions: why computational biology? Why a particular research area that you're applying? So think about that. Take some time to think about yourself, your background, and where you want to be, what makes you happy, what kind of research interests you or excites you. That would answer your question, I think. And don't let rejections demotivate you. You might get rejections from 1 or 2 places if you're applying for PhD or for postdocs when you're doing transitioning. But remember, you're looking for the right fit. If they don't think that your skills are a good fit in their lab, they might be looking at different roles of computational biologists, because computational biology is not one type. They can be data analysts, it can be pipeline development. So just looking at a vacancy, which is computational biologist, it might not be the right role for you. So think about that, try and apply in places where you think your interests lie and your skill sets will match as well. So my skill set matched with when I met Christine. It was very encouraging that she did not ask me what kind of background I had in terms of coding. Can I code in all the different languages that they use? She just asked me what your research background is and what my interests were. And that was enough for her to give me a chance to do PhD in her lab. I'll just take you through what work that I did, and that would hopefully make sense when I tell you about my transition to industry as well. So my research was focusing on protein family subclassification and protein function prediction. So this interest was obviously piqued by my biotech degree, where I was trying to purify proteins and understand their functional characterization. And I realized I can do a lot of it using computational tools. So that's where my interest lied. So what I was using during my PhD was using sequence and structure data to try and figure out the function of the protein and also identify functional sites. During the same time, because I did not have a solid computational background, I started attending the MSc coding courses that were available in the university in audit. And also I joined RSG UK at that time, which became a really enriching experience for me to try and talk to my peers. And I realized I was not alone. There were other people who were making the transition as me. They were coming from various different fields who were having similar struggles as me. So it was really helpful and became my support system as well during my PhD. And all of the people that I've met during those time have become really good friends and colleagues ever since. So what I did with my PhD was look at the cat proteins protein superfamilies, and my job was to come up with a functional classification that is automated so that we can find smaller groups of proteins which are likely to have similar function and structure. You may have heard about the PFAM family classification. Those are manually curated. You define the multiple sequence alignment by looking at their sequence and functions, and then you pull in sequences. What we were trying to do here was take all of the sequences in cat superfamilies and then automatically classify them into functional families, because we had realized with the increase in amount of data, we cannot actually use only manual approaches to try and do that. So initially, obviously, if I'm from a biology background, I can't from day one start method development. So the initial year, the first year, I spent on understanding protein function evolution and by studying certain superfamilies of interest, such as Rossmann folds and Tim barrels, how function evolves. And I did spend some time to try and appreciate what are the different ways this can happen, because these gave me ideas about how I can capture this in a computational approach. So I would try and say, take it slow, think about what problem you want to solve, and then look at what data you have and what are the steps that you want to take before you delve into method development or writing a tool, especially for people who are coming from a biologist background. So what I did by the end of my PhD was develop a systematic approach for doing this. And that was run for all of the cat superfamilies and is used for function prediction today. I'll not get into the details of it, but just mention that it's based on a hierarchical agglomerative clustering method where you split all of your sequences into very fine clusters and then try and build a tree and define an optimal partitioning. And this partitioning was based on how what do I say? It's based on comparison of residues which are conserved in one cluster and different in the other. And then you do a comparison and build up a scoring function based on what I had learned, how function evolves in particular superfamilies. So what we did then was try and use that classification method to predict function for unknown things. It's as simple as organizing data in a defined manner. Once you have an organization or classification system, you bring in sequences which match the same profile. And then if within your cluster, if at least one sequence has a structure or any function which is known, you infer that because you already have this assumption that you are happy with the clusters that you have created. So it's based on homology, but it has done incredibly well in predicting protein function, even when compared to machine learning approaches. Obviously, when we compared this when we added a machine learning aspect to this method, it obviously performed better. And we've been using that in the last 3 kappa function prediction methods. And the group has been working on this even after I've left the lab. Following my PhD, I did continue to do my postdoc in the same lab. I was part of the FUN-PDBE project, where we were trying to gather information on functional sites into a central resource. And I used the functional family information that I generated to try and interpret genetic variant, the impact of genetic variants on proteins. So if I can identify functional sites and then I see there are mutations around it, I can make better assessments of what impact that mutation or genetic variant would have on the protein. So that is what I was doing in collaboration with clinicians at the Francis Crick Institute. And that is where my interest in translational research also started. So this is where I realized that I was very interested in understanding disease biology and using computational tools to try and understand, make sense of genetic variants so that it can have more direct impact on patients' healthcare. So this is where I made my second transition, and I transitioned from academic research into a precision medicine startup. The reason why I chose to work for a precision medicine startup was because the focus was on patients and innovation. And I realized there was a drive to make an impact in a very short amount of time. And I also liked the interdisciplinary nature of the teams that they had. And also what appeals to me most is the collaborative environment, that they collaborate heavily with academic groups, pharma, healthcare providers, and even patient advocacy groups, such as the rare disease groups who are collecting sequence data and need computation analysts to try and analyze that. So that is where I made my second transition. And just to tell you a little bit about what we do at PrecisionLife, we know that disease biology is not as well understood as we would like to with all of the advances that we've made. We know that we tend to put diseases under an umbrella of a diagnosis, but we know that one medicine cannot treat all patients in a single diagnosis. All patients don't have the same symptoms, and all would not benefit from one treatment. And that has resulted in huge amount of healthcare costs as well. The most impact it has is on a patient's recovery time, because if you don't give a precise medicine in the right time, you're actually having an impact on that person's recovery time. So PrecisionLife aims to try and identify therapeutic opportunities which is more accurate for groups of patients, and it does not try and find one treatment for all patients. It tries to find which treatment makes sense for a subgroup of patients. And we also try and find biomarkers, patient stratification markers, and work on clinical decision support system tools, but all derived on the same basic idea that we want to find groups of patients who have similar phenotypes or similar genetic risks, or even environmental risks if we have the data. Just to give you a quick description of what that means, we know that population cohort studies try and identify one SNP, which allows us to identify, to segregate the patient population to people who have the SNP and people who do not have the SNP. But what PrecisionLife can do is find epistatic effects of not only 2 SNPs, but more than that, which allows us to identify groups of patients who have these combinations of SNPs, which would have a different impact than the other group, which probably don't have that signature. And this is how we identify the patient stratification network and derive our insights based on that. So these are generally derived from genomic data, because that's what is available for most of the patient data sets, but we also can combine electronic health records, epigenetic information, transcriptomics data, or proteomics data, because we know that most chronic diseases are not caused by one gene, they are caused by multiple genes, and not only they are associated with genetic risk factors, but they can have risks which is environmental or a combination of both. Very quickly, GWAS gives us one SNP, but we are able to identify a combination of SNPs, which allows us to identify not just one gene which can have an impact, but potentially identify which pathways or which metabolic networks might be affected by a particular patient. I'll not get into the details of this example, but use the leftover time to talk to you about my reflections of the journey that I have had. Finding my niche was challenging, but that is where I've realized that I am a biologist at heart, and I can't be the best programmer or have the most sophisticated coding skills. So I've found a niche for myself that I know that my strengths lie in data analysis, thinking about the experiment, thinking about the experimental design, and having a broader picture of what data I want to bring in, what that means, and what I can talk to with computational scientists who can do a better job of developing pipelines and more sophisticated ways of improving performance. So finding your own niche my own niche has been very helpful in building up my confidence that I can't be the best in everything, but I can identify what is my biggest strengths and go with that. And along the way, I have learned a lot of the things with the people that I've worked with. They keep on telling me new things, and I keep on learning that. And again, knowledge can come from different places. It can be an intern who's working with you, who's found this new cool way of doing the same thing that you've been using 10 lines of code, and it can come from your mentors and supervisors as well. Another thing which has been instrumental in my journey has been choosing the right fit, finding the right role, the right mentor, and the right team for yourself. And it need not be always the best opportunity which at that time appears to you. You just need to think about whether it's a place where you want to work. It can be your PhD, it can be your industry job, wherever. You need to think about the people that you're going to work for, whether there's a culture which is suitable for you, whether you would have a voice that will be heard, and you will have the guidance and support from your mentors if you need help. So if one of the crucial things is to ask for help when you need it. Often we tend to not ask questions because we think that we should be knowing that. But having an environment where you can ask questions is very, very important. So find the right role and the mentor and the team that you want to work with, and that would make life much easier, and you can actually focus in your work that excites you. Finding courage and support, that has been quite a journey for me during my PhD, because to take risks and challenges where you're doing a transition is quite hard. And I always suffered from that feeling that I'm not a computational scientist, so I don't know how to do this thing or that thing or whatever. But finding the right support really helped me. So I've received support during my PhD from the supervisors, senior members in the lab, people who I've met in conferences and symposiums who have answered my questions. Talking to peers, as I mentioned, RSG UK was really helpful for me to meet my peers and talk to them about their research and learn new things. Also talking to people from different disciplines. So I used to stay in a student accommodation where we used to all talk about our research, and we realized how much we could learn from each other's disciplines, even if they are really different. Transition from academia to industry was another daunting task, because I thought I did not know what the industry would be like, what should I be expecting. But what helped me was attending talks, conferences, and webinars that are available to join. These days, you can find them on Twitter if you follow some of the student groups across the world. Also, one of the key things which helped me boost my confidence was attending data dives or biohackathons. You also have those for the ongoing COVID crisis, where you would be able to join Slack channels where they have the data and they're just inviting people who are interested in exploring the data. Join those, talk to the people if you're interested. You'll learn a lot of things and realize that these people are also coming from backgrounds very similar to yours. So don't let it demotivate you if you think that you don't have the skills. The skills will come with experience. Just go out there. And support from manager and colleagues as well. Finally, I wanted to mention that it is very crucial to understand the data and the tools and not only focus on the results. You can get brilliant AUCs, that's fine, that would help in benchmarking results of the paper, but what I found was I really needed to understand the data and the problem to understand whether the AUCs are correct and they are exactly reflect what I wanted to come out from it. Because I can develop a tool based on the training and the test set, but would it work for everything else which is out there? Because my end result is to try and find genetic variants which has probably never been encountered before. So I need to find whether my method is generic enough as well. So keep on asking questions. Do I have the right data? What was the experimental design to generate the data? Talk to the people who generated the data. What are the confounders? Think about all the possible confounders and go through them. See whether you've checked through that and focus on solving the problem. Finally, I was told to talk a little bit about transitioning into industry and what skills I think the industry looks for. I'm not an expert in that, but I'll try and highlight some of the things that I think is useful for people who are making that transition to highlight, because often we feel that it's in my CV and they will see it, so I don't need to mention or highlight it. Don't do that. Try and highlight as much as possible. So identifying the right role for yourself, as I said, computational biologists can be of different types. See whether the role is actually fit for you. For example, I would probably have never applied for a pipeline development role for NGS because that's not what I do, but the job title would have been the same, but I would have applied if it was interpretation of the computational biologist. Not that I don't do it myself, I do it, but that wouldn't have been the only thing that I would have applied for. Recognizing and highlighting relevant experience. Most of the times, the job applications would not be an exact match of what you would have done, but try and find similarities of your experiences that can have a positive impact on what the companies or the job roles defines. Try and find similarities, what things you have done before. You may not have worked on RNA-seq data, but you may have worked on extraction of RNA. Highlight that experience because that's useful for the person to know. Any project planning and managing or communication experience you have, highlight that. By project planning, you don't need to have a job title called project planning, but we do that. Any person who has ever picked up a pipette and tried to do a biological experiment, you have done planning. And be open-minded and collaborative. You would be working in an interdisciplinary environment where you would be asked questions from different viewpoints. So keeping an open mind about things is very, very important. That helps to obviously solve the problem at hand and not be have some biases which come from a certain discipline. Again, positive attitude, willingness to learn. That's true for everywhere, I think. That's not unique to industry. And finally, being well-informed and curious. Everybody likes to know that you've looked at them, you are actually interested in them, and you're curious. That means that you're not afraid to ask questions. If there's something that you know of, you have knowledge about, you would ask questions. That's what people would want, because people would need, you would almost like represent your experience. People would be looking up to you to try and get insights of what you think we should do. So everybody should have like opinion is like invited almost. So ask relevant questions. And just wanted to leave you guys with, if you ever thought transitioning is difficult and maybe you are the first person doing it, don't think so, because even the experts have done it and they have gone through the same challenge. So my supervisor had done her PhD in experimental science and then moved on to doing computational biology. Professor Dame Thornton, she was a trained physicist who had moved on to do computational biologist. So if they can do it and they have inspired us to follow their footpath, I've been inspired by them and they're my role models. So I would just encourage you to listen to them as well. And if at least it tells us that it's possible, right? To gain success by changing fields of if you're following your interests. Finally, I would just like to acknowledge my universities, my mentors, my colleagues, and everybody, because all of them have played a part in me understanding where I want to go. And with that, I've come to the end of my presentation. So just let me know what questions you have.
Thank you so much, Sayoni. It was fascinating, especially to see at the end Janet's face. I worked in Janet's lab for a year or so. So you're absolutely correct that the mentors of today's time in this field actually are multidisciplinary in truly speaking. Now, before beginning the questions that students have asked, I have two questions to you. One is that this, I think, people with biology background have an undue advantage over, and I think you may help with this question. Do you think people with biology background actually know better what to analyze and what to look for than people from computer science background?
This is going to be a very biased answer, I think. I'm not sure whether I can say that, because as I just showed you, my supervisors obviously came like, if Janet has come from physics, I cannot say that she doesn't know what to look for. So
Obviously.
I think what makes the difference is often it is, as I mentioned, alluded to the fact that each discipline has its own bias, which is implicit. It's just how we've been trained to look at certain things, because it's a natural focus for different disciplines to look for certain things. Being a biologist, I'm more interested in, when I see a result, I'm like, which protein is causing this effect and what genetic variance is causing this effect. Let me see if the data fits in. I mean, I already know that there's going to be huge complexity. That's what I've seen. It takes some time to get used to the complexity of the biology systems. And I think if a first-year computational scientist and a first-year biologist who are transitioning into computational biology sit together, I would be biased to say that probably the biologists would have more appreciation of what are the different aspects which may have an effect on the data. But the computational scientists would understand after trying to build the models in time, basically.
Sure.
So I think that's the difference, because the computational scientists would focus on AUC and say, that these are the dominant effects. But then the biologists would say, I want to look at which are not the dominant effects and tell me about that. So I think it's just the kind of questions we try and think about as different disciplines. And that's why the best work happens when both of them are working together to try and answer the same problem. So
second is, even in my lab, out of my first 3 PhD students, first 2 were from biology background, hadn't written a single line of code, and they transitioned into computational biology lab. I don't know why, but still they're figuring it out. But from your point of view, how did you overcome this general fear people have over programming or analysis? Or, I mean, people are, I think the students that are asking questions, they would, I guess, would be transitioning from Excel data, and how to actually overcome this fear of lines of codes. What motivated you?
Thank you for the question. I'll actually try and tell you a funny story. When I was trying, as I mentioned, when I was doing my MS research, I thought I'll gain my bioinformatics experience. Then I went and spoke to Jairam Ramesh that, Professor Jairam, that I wanted to work in your field. But then it was the department clash, like, oh, you can't have a project from a different department. And okay, things didn't work out. So I started looking at bioinformatics tools. I tried to refresh my memory, what I'd learned during my biotech course. I actually ended up giving the first BINC examination. I think that was 2010 when the BINC examination was first started. Having only written Perl programs which allowed you to swap palindromes and read up a sequence from a file, I actually gave the exam. I passed the theory because that's what we do. I got 0 in my practical test because I knew how to open a PDB file, but I did not know how to do Ramachandran plot. So I could find all the coordinates and everything. And I accepted that. So I was like, I don't know this. But after the test, I read up how I actually wrote a paragraph what Ramachandran plot is, and I wrote a pseudocode of I'll do this and this and this. So I think coming to the fact that accepting that I don't know this, but it's not like if I have the chance, I won't be able to do it. Probably that was the main thing which was driving me. So my friends were crazy. They told me I was crazy because why are you giving this BINC examination? There was no point in doing that. You've not even done coding. But I just wanted to test myself. What do I know? Let me go and see and just accept the fact that I'm fine, that I don't know how to code. But within this 6 months, I again finally knew how to work with PDBs and been working with them ever since. So I just would encourage people to take that push. It's fine to fail in 1 or 2 attempts. It's not like I got through all of the PhD offers, but I'm glad for the offer that I got because that was the perfect fit for me. I did have an offer from another reputed university where I did not feel the environment would be perfect for me because it probably wouldn't, it would probably be expected that I know certain things, even though I've got the offer. So I chose an environment which would allow me to get that time to learn stuff. So do yourself a favor, find an environment where you think you would thrive, and it's fine to look for a certain time. I lost 1.5 years, you could say, when I'm doing my master's because I'm not using that. But I don't see it that way because I learned a lot about experimental design at that time. So just take your time, follow your dreams. If you're really interested, follow it. If you think, also embrace computational biology no matter what. You would need to do a BLAST. I mean, my friend who's a synthetic biologist has learned bioinformatics tools and it's incredibly useful. So embrace computational tools no matter what. It's almost like mail changing into email. So any biologist would need to use some amount of computational tools, no matter what.
Right. At least of the 21st century research, it is going to be a prerequisite, at least a little bit. Okay, so one question was interesting is textbook biology is different from real biology. And it is really challenging for me. How to cope with this challenge?
Look at data, I would say. There's no dearth of data now. How would I say? You have Human Protein Atlas, you have UniProt at your disposal, you have PDB at your disposal, the CATs, all have highlighted the diversity of data that's out there. That's probably not something that you will see in textbooks because nobody has time to rewrite textbooks, everybody's writing. And the ones which are being written are not available very easily. They are very costly, the ones which come out. So I would say try and join those bioinformatic exercises. Rosalind has them. Where you start coding, they have simple problems and you can code there. Join any of the COVID efforts if you're really interested. They have sequencing groups who are putting all of the sequencing data out there. Just talk to them, just follow the Slack channel and just see what they're thinking about. It's easier if you don't start with a new problem, if you try and think about the problem that you have. If you're looking into a particular protein, look at sequences across species. If you're looking at genomic sequences, try and look at, if you're looking at a particular cell, don't look at that cell only, look at everything else. So I think identifying the diversity of data is quite important for biologists venturing into that because there is no rule. Dr. Leelavati has put that before. There is no single rule, but we are just trying to find patterns in the data that is available to us. What the method, the first version of the method that I wrote, there was no proteomic sequence surge at that time. That was 2014. And in 2 years, we started sequencing all the different proteomes. And then suddenly that algorithm did not make sense. We had to add it, like make improvements. So we are always seeing a snapshot, and that snapshot is getting bigger and bigger with time. So we're learning every day, basically.
Absolutely. And I think one message that is completely clear from both of the talks of you and Dr. Leelavati is that understanding computational biology as being a geneticist or being immunobiologist, it's a vast field where actually we put on a lot of hats. It's just that anything that is relating to biology and involves using a computer could be a computational biologist, but I think the the finding the niche, as you said, is a vast field where one can always have their space. Well, with this, I have no further questions, and I think the students till now have no further question. There was one question with Dr. Leelavati, if she wants to take it up, being a student from electrical engineering and with all the computer science background, core electronics domain, do not have enough good experience in programming algorithms. I'm confident with scripting using Python, Shell, TCL, and Perl. And basically had a basic understanding of biology. So how difficult would that person feel in a field like computational biology?
So I had an intern who came from the exact same background, a summer intern from electrical engineering. I think what you really need is logic, because if you're doing scripting already, you're right there. I mean, you're pretty much there. You're doing already programming. So don't, lessen your achievements, right? I mean, it's already there. A few exercises in programming might work, but depends on what you want to solve, right? You don't really always need to do complicated programming for the problem. It depends on the problem that you're trying to solve. If you want to write BLAST from the beginning, then yes, you're going to be, it's going to be painful to do. But if you want to do a small snapshot to see if dynamic programming you can understand, it's fairly easy to do. Just a couple of for loops, and that's it. Right. And what was the question again?
Basically, an electronics person, electronic engineering person being interested in computational biology.
Yeah, I think, like I said, from any background, we've seen people transition into doing. I think if you are passionate about the problem, you'll figure out a way to solve it. So don't worry about what background you have, what decisions you made in high school. Doesn't matter. So I think, and that is something that we need to stress. Nothing, just don't stick to the background. Yeah.
I know a lot of electrical and electronic engineers who transition into neurobiology and doing computational biology work in neurobiology. Fascinating work they're doing. Great. With this, I think if there are no more questions, then I would really like to thank Dr. Leelavati and Dr. Sayoni for an invigorating Sunday afternoon and during these COVID times. And at least it's good to see at least few faces, hear science, and hear exciting experiences. Finally, from my end, again, I would like to thank ISCB RSG-India. I guess you guys already, the core team, you guys already realizing that how much this society has touched lives, because you have Dr. Leelavati talking about getting funding for travel, Dr. Sayoni, who has been a secretary in RSG UK, and I also was associated with. So keep up the good work. And I think this platform and these questions and these seminars are extremely important when it comes to exciting young minds. I'll hand it over to Manisha in case you have any last words. And thank you for inviting me.
Thank you. I had a lot of fun.
Thank
Manisha,
you.
Manisha, your mic is mute.
Yeah. So thank you, Dr. Ray, for accepting our invitation for chairing this session. And thank you, Dr. Das and Dr. Narlikar, for giving such an interesting talk about your own transition to computational biology. Now I give it to Shankar. Shankar?
Yeah. So on behalf of RSG India, I take this occurrence to thank everyone who joined us today. We've been fortunate to have renowned scientists from academia and the industry. And I must remark a proficient sense of gratefulness to all of our guests for sharing with us their experiences, despite it being a Sunday. And a lot of relatable input regarding how this field is interdisciplinary and how thinking with an open mind or a different perspective would actually help us solve problem was shared by Dr. Leela. And Dr. Sayoni's experience on how she audited courses would have given a sense of hope to all the attendees. I hope this would have inspired all of us to put the effort and not get frightened to make this transition. I'm sure all the participants of this meeting found it useful to understand the scenario and make better decisions. And finally, I thank our chair, Dr. Arjun, for taking the initiative of hosting. Thank you.